Okay, so the glomerulus increases blood pressure by forming narrow branches. The pressure is maintained by the narrow efferent arteriole, which restricts the outflow of blood from the glomerulus. The expanded end of the nephron forms a cup that accommodates the glomerulus, known as the glomerular capsule. The efferent blood vessel associates itself with the other regions of the nephrons for selective reabsorption. High pressure is generated in the glomerular knot, and fenestrations, gaps between the cells that form the glomerular blood vessel, create a path of low resistance out of the glomerulus. So the fenestrations can be seen right here as this little space in between. Okay, so, and then the basement membrane, which is right along here, it is the filtration barrier, and cells and large plasma protein macromolecules cannot pass through this structure. So ones like that are this big can't go through there because the space is too small, but smaller molecules like this one can. Okay, and podocytes, which is this right here, they are in the inner membrane of the Bowman's capsule, and the podocyte extensions create gaps for the filtrate to pass between the cells. And that's the purpose of the glomerulus and the glomerular capsule.
Tubular reabsorption occurs at the proximal convoluted tubule. In tubular reabsorption, molecules and ions are passively and actively reabsorbed from the nephron into the blood of the peritubular capillary network. The plasma proteins and salt regulate the osmolarity of the blood. Sodium ions are actively absorbed and chlorine ions follow passively. Here the salt ions are shown in green. When the salt ions are reabsorbed, the osmolarity of the blood compared to the filtrate increases, and so water, shown in blue, moves passively into the bloodstream. Glucose and amino acids, shown here in pink, are also reabsorbed by active transport into the blood. The wall of the PCT consists of single-layer ring cells. They contain mitochondria which produce ATP for active transport of materials. The microvilli increase the surface area for reabsorption. About 80% of salt ions, 99% of water, and 100% of glucose in the filtrate is reabsorbed into the bloodstream. Substances not reabsorbed enter the loop of Nephron.
The function of the loop of Henle is to create a salt bath with the surrounding renal medulla fluid. After the filtrate leaves the proximal convoluted tube, it enters the descending portion of the loop of Henle. A lot of water still remains in the filtrate along with remaining dissolved solutes like sodium, potassium, chloride, bicarbonate, and hydrogen, represented by the green molecules. Water is represented by the blue molecules. The descending portion of the loop of Henle is permeable to water, but impermeable to salt ions. Water leaves the tubule and gets absorbed into the bloodstream. The volume of the filtrate is reduced, and the salt concentration in the filtrate increases. The filtrate goes through the base and ascending portion of the loop of Henle, which is impermeable to water and permeable to salt ions. Sodium ions are pumped out through active transport by the use of ATP into the intercellular fluid of the renal medulla, which as a result is a hypertonic region. Even though water moves out of the descending portion of the loop of Henle, the filtrate that is left is still hypotonic, meaning it still has a high water content compared to the renal medulla. Next, the filtrate enters the distal convoluted tubule.
The distal convoluted tubule is composed of cuboidal epithelial cells that have numerous mitochondria but lack microvilli.
Because of its structures, it is more likely to help move molecules from the blood into the tubule, a process called tubular secretion. In tubular secretion, substances such as hydrogen ions, potassium ions, creatinine, and drugs are removed from the blood and the peritubular network and added to the tubular fluid by active transport from the blood. DCT also plays an important role in acid-base balance by acidifying the urine. Normally, it is relatively impermeable to water. However, in the presence of antidiuretic hormone, ADH, its permeability to water increases.
We now move on to the collecting duct. The filtrate enters a nearby collecting duct. So what is a collecting duct? Well, collecting ducts are a series of small tubes inside the kidneys, and it's the final component of our fluid filtering process. The filtrate enters one of the collecting ducts. This collecting duct is differentially permeable to water. The permeability depends on the presence of ADH, also known as antidiuretic hormone. Now, in our last unit, we learned that ADH is secreted from the posterior lobe of the pituitary gland and circulates in our bloodstream. In the kidney, the collecting duct extend into the hypertonic medulla oblongata through here. If ADH is present, the collecting duct then becomes permeable to water, and water moves by osmosis out of the collecting duct and into the surrounding medulla intercellular fluid, as shown by the movement. If ADH is not present, the collecting duct becomes impermeable to water, and water will stay in the collecting duct with the other waste solutes, and the urine becomes more dilute. Osmoregulation takes place nearly entirely in the collecting duct. What is osmoregulation? Osmoregulation is the body's response to mechanisms which attempt to maintain homeostasis levels of water. To summarize, if the concentration of blood plasma is high, osmoreceptors in the hypothalamus detect this and send nerve impulses to the pituitary gland to secrete ADH. The ADH is sent to the collecting duct so that more water can be absorbed, resulting in concentrated urine. When we drink more water, the pituitary gland secretes less ADH, so less water is absorbed by the capillaries and blood plasma level is therefore lower. The volume of water eliminated depends on three things. The total volume of water ingested, so that includes liquids and the liquid and solid foods. The perspiration rate, which depends on exercise and temperature. And finally, the ventilation rate, which is your activity level. Kidneys can therefore regulate pH by removing the excess acid or bases in the urine and restoring the bicarbonate concentration in the blood to normal. Now, the differences in the concentrations of proteins, glucose, and urea between blood plasma, glomerular filtrate, and urine. So the products move in this sequence. Blood plasma, glomerular filtrate, and urine. Well, why is that, you ask? Well, during this process of filtration, urea is eliminated from the body through the urine, but not all of the urea that leaves the collecting duct is taken up by the loop of Henle, so there's still 50% of total reabsorption. Glucose is reabsorbed from the filtrate back into the blood, all of it, and proteins are found in the blood plasma, but they do not move into the glomerular filtrate. If proteins are found in urine, then your blood pressure is too high and there could be damage in the Bowman's capsule. Therefore, as I have summarized in this chart, we go from blood plasma, glomerular filtrate, and urine.